Hello, everyone. Welcome to the first of our outreach lectures to aimed at students from rural backgrounds. And hopefully this will be the first of many. Of course, when it comes to doing outreach in places that we're not familiar with, we do have a lot of technical things which we have to deal with, and I'm sorry about that. And yeah, so I'd just like to give a bit of background about the Bangalore Life Science Cluster. We are a group of three institutes: the National Center for Biological Sciences, the Institute of Stem Cell Science and Regenerative Medicine, and the Center for Cellular and Molecular Platforms. And NCBS works on fundamental research. InSTEM works on translational research, and CCamp is a startup incubator. So it is a very it's a great campus to be on, where fundamental research, translational work, and applied biotech startups are all basically in the same campus. And it makes for a lot of amazing diversity on campus, and a lot of interesting science happens here. And today our speaker is Dr. Deepa Agashe. She works in the adaptation lab of NCBS. She heads the lab, and she is interested in integrating ecology and evolutionary biology with molecular evolution to understand the processes governing adaptation. She also favors experimental evolution approaches to understand adaptive dynamics during bacterial genome evolution, the ecology and evolution of diet specialization and expansion, and population differentiation and divergence. And what is more important, त्यांना मराठी येतं. आणि सो मोस्टली तर टॉक आहे जो तो इंग्रजीमध्येच असणार आहे. बट स्टुडंट्सला तुम्हाला कुठले प्रश्न त्यांना मराठीमध्ये विचारायचे असतील किंवा हिंदीमध्ये विचारायचे असतील, तुम्ही ते विचारू शकता. आणि प्लीज 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 प्रश्न विचारा. म्हणजे ग्रामीण भागाचे मुलं, किंवा म्हणजे आता तुम्ही ज्या कॉलेजमध्ये असता तिथे युजली मुलं फार शांत बसत असतात. तर इथं एक क्वेश्चन आन्सर क्यूए बटन आहे, तुम्हाला दिसेल. तिथं टाईप करून तुम्हाला जे क्वेश्चन्स वाटतील तुम्हाला ते मराठीमध्ये लिहायचे असतील तर मराठीमध्ये लिहा. त्याचे म्हणजे इंग्लिशच्या स्क्रिप्टमध्ये मराठीमध्ये लिहिले तरी चालेल. आम्ही सगळे प्रश्न जे आहेत तुमचे ते सोडवायचं, आणि त्याचं उत्तर द्यायचं आम्ही प्रयत्न करू. आणि टॉक पण तुम्हाला जिथे कुठे वाटलं की थोडंसं एक्सप्लेनेशन जास्त पाहिजे, प्लीज तुम्ही ते Q&A मध्ये वगैरे टाका, आणि आम्ही ते सोडवू. प्लस युट्यूबवरती पण हा टॉक आहे, म्हणून तुमच्या फ्रेंड्सला वगैरे तुम्ही पाठवा. आणि तुम्ही जितके प्रश्न विचारू शकता तेवढे मी जास्त टॉक्स आणि पुढे ऑर्गनाईज करू शकतो तुमच्या सगळ्यांसाठी आणि भारतात सगळीकडे. त्यामुळे तुम्ही जेवढा रिस्पॉन्स देईल तेवढं माझ्यासाठी पण चांगलं आहे, आणि तेवढं पुढच्या पण लेक्चरसाठी चांगलं आहे. So with that, I'll just hand it over to Dr. Abhay Khandagay, who's the principal, and he will say a few things. So, yeah. Dr. Khandagay.
Thank you, Chandrakant. On behalf of Pune District Education Associations, Professor Ramkrishna More Arts, Commerce and Science College, I wholeheartedly welcome Dr. Deepa Agashe, Dr. Mahin Ali Khan, who are very much on this platform, which is in a way unique for our students. Though students are quite accustomed with Zoom, but then whom are they going to interact with is something that really matters. And today they have eminent personalities with which they will be interacting. I hope they will be interacting because this is something which has to be thought in view of the new normal that is slowly getting set up in our country. How are we going to sustain the situation? How are we going to pull on with it? Because being a principal at this point in time, I am facing a lot of challenges, and I'm enjoying the challenges, of course, like how to link up with students, how to call them, how to arrange the lectures. And when I see the attendance reports of many of my teachers, what I get to know is students, they join in, and unfortunately, they have to leave because of the internet problem. And then the in and out, it goes on for at least 7 to 8 times in 45 minutes of lecture. So then that, really shakes me up. Like, what is it that I should do for it? So I'm constantly in touch with several agencies to give the best of them. And I'm trying to give them 24 by 7 some material which they can download and use it. And our DBT overall coordinator, Dr. Rashmi More, is very proactive in organizing lectures, lecture series, hands-on workshop, visits, internships, and whatnot, especially for this group of students. This is especially for Dr. Deepa Agashe, that the students whom you will be addressing are, uh, like 90% of them are from rural background. There are only 10% who live in the so-called fringe of the urban area, but they basically come from a very remote place. There are so many who live here, but they live here in some private hostel, or sharing a room on court basis or something of that sort. So then we are trying to keep these students in the mainstream. And then what we basically feel is these students, they should listen, and they should develop the skill of listening. Once they develop the skill of listening, they are going to be really wonderful students, because I have seen them. They come to me with 2 mark list, 3 mark list, 40%, 45% in 10 plus 2. But when they get graduated, most of them get 75 and above. And that is what I have attained. And maybe this is the point why my college has been awarded with A plus grade by NAC Bangalore. So it's not like I'm boosting myself. It's only that my students, they can really do well. Only thing is we have to keep them, in the mainstream and on the right channel. And I'm very happy to have a dedicated teacher and staff and all my zoology staff. There are so many staff members. They have also joined, and all of them, they constantly keep a track record of the students, like what are they thinking, what are their abilities, what are their skills, what is their like inclination towards, and accordingly we try to promote them. So without taking much of your time, I would like to hand over the session back to Chandrakant, who was my colleague and now is a good friend of mine. So back to you, Chandrakant, please.
Yeah. Okay, yeah. So Professor Khandagai gave me my first teaching job many years ago. So that's how we know each other. So yeah, so I think we should begin with the talk. So Dr. Deepa, could you please proceed and begin with your lecture?
Yeah, so I'll say one quick thing. Chandrakant, if you don't mind, can you keep an eye on the questions because I can't see them
Yes, yes.
while I'm playing the
I'll make
slides.
sure. Yes.
And please interrupt me if something is,
Yeah.
I mean, the questions clearly say that, something is not clear. I'm
Yeah.
happy to explain in the middle.
Yes.
So I'll get going. Let me share my
Yeah.
screen.
Yeah.
Okay. All right. You can see my first slide.
Yes.
Okay, great. Good. So welcome everybody, and I am very happy to be here, speaking to you in this session. And one second, I'm just going to make sure that my own face is not in front of me too much. Okay, there. Yes, I'm really excited to be able to talk to all of the students who are gathered here, and I hope that I can tell you why I'm so excited about some of the work that we do. And this is largely to do with evolutionary biology. So I will speak in English, but I'm also comfortable speaking in Marathi. So if you would prefer that I speak in Marathi, I'm also happy to do that. So please just interrupt at some point or put your questions in the chat box, and I will do my best to explain in Marathi. I don't know the technical terms in Marathi because I learned these scientific terms in English, but I can certainly explain in Marathi as well. So welcome again, and I'm going to take you through a bird's eye view of what I mean by adaptation and what do I mean by saying we're trying to unravel adaptation, right? So I'll start with some interesting videos because that's always fun. So the first question many of you are thinking about is, what is adaptation? right? What do I mean by adaptation? What do scientists mean by adaptation? So I'm going to share some videos here with you. Hopefully you can see. So this first video is showing you a cheetah that's running, right, in the plains of Africa. And you can see it's running at this top speed. And I'm sure that most of you have seen some such video in the past, right? So this can be considered an adaptation, right? That it's running very, very fast. And the prey it is catching are also running fast. And that's why this evolved this adaptation to be able to run as fast as these prey. The second video you start seeing here now is a fish, and this fish is swimming around with its babies, right? It's oh, and look at what it did. So this fish, it didn't eat its own babies. What it is doing is it's providing shelter in the mouth, which is the safest place from other mean predators that are hanging around, right? And so this little school of small baby fish will often go and hide in their parents' mouth for protection. So this is an interesting case of parental care, which of course as humans we're familiar with parental care, but it's very, very common in the animal world as well. And it's very, very fascinating, these kinds of adaptations that seem to have evolved in different species. The third video here that I'm playing is an ant, and it's walking in the rainforest in, well, I don't know actually which rainforest, but somewhere in the Amazon perhaps. And as you see, it's doing something a little bit strange where it's now just resting in one place. And maybe we'll watch a little bit longer. So time passes now, and you see this fungus that has grown up. And now if you watch closely, there's a stalk coming out of the head of the ant, right? So this is a very, very fascinating behavior where this fungus, once it infects an insect host, changes the behavior of the insect, okay, and makes it go and clamp its jaws and attach itself to a particular position in the canopy, such that what scientists think what's likely is it's an adaptation to make use of the optimum humidity and temperature regime. And then the fungus slowly kills its host and grows out of its body. And those black spores that you see at the end are the ones that will now propagate the right? So that's a really fascinating adaptation that a parasite has evolved to be able to infect maximum hosts and get around really, really fast. And finally, the last video that you see here is of a bee. And let's again watch it for a little bit. So this is a flower, it's an orchid. I think many of you have probably seen or heard of orchids. And these are beautiful plants. And what they do is they mimic the scent or the appearance of many kinds of females of these different bees or wasps. And they attract the male bees who think they're going to mate with a female. And instead they get stuck in there in the nectar. And then the flower, the shape of the flower forces the bee to go outside once it's stuck in the nectar through a narrow passage. And on the way out, it makes sure that there's a pollen grain or two which is attached to the bee on its way out. So effectively, the plant is fooling the pollinator into pollinating itself, right? So this is again perhaps an interesting adaptation where the plant is mimicking something which the insect has evolved to recognize as being important for its life cycle. So these are all cases which I think many of you can think of as many different aspects of these behaviors or these observations you can say of our adaptations, right? But I'd like to dig a little bit deeper than that. So in evolutionary biology, we actually call two things adaptation. One is the process itself through which these adaptive behaviors evolve, right? Or adaptive functions evolve. So for example, we autism is a blind adaptation, right? So why is the cheetah able to run so fast? How does the plant evolve these volatile chemistry and the compounds that allow it to attract pollinators? What is the rate at which these process takes place? Is it very fast? When is it fast? Why is it fast? What are the other factors that allow adaptation to be really fast? So those are the questions that we can ask there when we're trying to understand adaptation as a process. Second way we can think of adaptation is as an outcome. And that is largely how we typically think of adaptation in a everyday sense, right? Where we think of the outcome of adaptation and we say, well, what is the benefit of this particular behavior? Is it actually beneficial? What are the consequences of this behavior? Is it an adaptation? And that's how we define an adaptation, right? So technically speaking, we would say a particular behavior or a particular function is adaptive. It really has a measurable increase, it results in measurable increase in the ability of that organism to either survive better or to reproduce better, right? And everything else in evolutionary biology typically sort of narrows down into these two things. No matter what we think about, whether it's a particular gene, it is a particular mutation, it is a particular trait, a function, are you running fast, are you running slow, all of it is only relevant in evolutionary biology if it affects your survival or if it affects reproduction, right? And so different evolutionary biologists will approach the question of adaptation in these different ways. Either they can think of process, they can think of outcome, and then try and understand what's going on. And so when I say unravel, what do I mean by unravel? I mean, so I guess the direct translation of unravel is ulgadna, Marathit, right? So खूप कॉम्प्लेक्स गोष्ट आहे तर ती आपल्याला सोडवायची आहे, गुंता आहे तो आपल्याला सोडवायचा आहे. How did this thing evolve? What is underlying it? Can I make predictions? If I understand how this works, can I say how that will work as a result? Those are the things that we're interested in. Maybe I'll pause for a second and ask if people have any questions.
I don't see any questions here yet. पण कुणाला क्वेश्चन असतील आता तर ते हातवर करू शकतात. एक रेज हँड आयकॉन तुम्हाला दिसेल तर तिथे तुम्ही हातवर म्हणजे करू शकता आणि मी अनम्यूट करतो तुम्हाला आणि तुम्ही क्वेश्चन विचारू शकता.
Okay, all right, ठीक आहे.
Okay, Ashish has raised his hand, so we'll just ask Ashish.
Sure.
So Ashish,
मॅम, हॅलो, आवाज येतोय माझा?
हो येतोय.
सर ते थर्ड वाला व्हिडिओ आहे ना अडॅप्टेशनचा, ते समजलं नाही.
थर्ड वाला व्हिडिओ म्हणजे त्या मुंगीचा?
हो, मुंगीचा.
So त्याच्यात there's this parasite which is a fungus which goes and infects the ant and then makes the ant behave in such a way that it goes and dies. Instead of going out and getting food and going with its colony, it doesn't do that. It goes outside, it chooses a spot slightly up above the ground, and it uses its jaws to just sit there, and then it dies there, right? Because it's not eating anything, nothing is happening. It just sits there, it dies, and the nutrition inside its body is used by the fungus to then reproduce and produce that big stalk that came out, and then produce spores. And then the spores can be dispersed by air or by other mechanisms. So that we think is an adaptation on the part of the parasite to evolve, manipulate the behavior of the ant such that its reproductive success is maximized. Is that helpful?
Yes, thank you, ma'am.
Okay, sure.
We've got one more question.
Sure.
Suhas Desai is saying, how much time does it take for a mutation to appear or occur? Can it be instantaneous? So
that's a good question. Okay, so the way we measure this, saying how often do mutations happen is, I mean, we measure it as mutation rate, and typically we will express, or the units of mutation rate will be number of mutations per base pair per generation. Okay, people can use different units as well. It can be per base pair per genome as well, but per base pair per generation is the most frequently used in evolutionary biology, at least. And so that rate actually varies across organisms quite a lot. So it is very high in a, I'm sure you've heard about the coronavirus, for example, and one of the reasons people have been worried about coronavirus and its evolution, and whether it can evolve really fast so that a vaccine will not work anymore, and so many questions that come up. They all come up because viruses evolve typically fast, and that's because they have very high mutation rates. Okay, so mutations occur very fast in viruses, typically within a few months' time. Now, for the coronavirus, the estimate is we should see 2 to 3 mutations that appear in populations and spread in populations within this 2 to 3 month time span. For bacteria, the mutation rate is a little bit smaller or a little bit lower than what you see in viruses. And in other animals like humans, the mutation rate is even lower typically. Yeah. So, so the rate actually changes quite a lot. In human cells, we do expect that every human being has many mutations compared to each other. Okay. Because in our so in the time frame that it takes, the number of generation it takes to go from a fertilized embryo to a full human being, a grown-up adult, our cells have undergone many cycles of replication, right? And how do mutations occur? Mutations occur either because of external damage directly. So if you, for example, are exposed to UV radiation for a long time, you know that you can get skin cancer. That's because of mutations that occur in your skin cells due to the UV radiation that damages the DNA inside your cells. But mutations also occur when your cells are trying to fix that damage. Okay, they're trying to repair the damage, and sometimes they make mistakes while doing that repair. Okay, so that's another way in which mutation can happen. A third way mutations can happen is during the process of replication itself. So you know that whole DNA is copied every time cells replicate, right? So during that copying process itself, the enzymes that do that copying are not perfect. They make mistakes. And so that is another way in which mutations can happen. And so because mutations can happen in these different ways, the precise rate of mutations or exactly when mutations will occur is very hard to describe, right? We can say the average rate of mutations for a given species in a given condition, in a given environment, and so on. But it's very hard to pin down and say, okay, I can predict exactly when the next mutation will happen. That is very hard to do for any given organism. Does that answer your question?
It was a written question, so
Okay.
but I think that was, yeah.
Okay, all right.
You can go on with the talk.
Okay, all right, fine. So then, so how do we start studying adaptation, right? And so today I want to not tell you so much about exact results that we have found in our experiments or in our work, but I want to give you an understanding of how we even start approaching this problem, right? So what do we need? We need variation, right? So this is a very nice poster. Google for it, I find this, of different eyes, right, of different organisms. So you can see a human eye there, you can see a frog's eye, you can see a bird eye, you can see a reptile eye, there's a chameleon eye, right? So there are many different, there's a lot of variation in what eyes look like, right? Even though they all serve the same function, that they want to capture light, create an image in our brain, and then we can process the world, right? We can respond to the stimuli, we can respond to the world, we can see where we're going, all of that. So all these eyes are serving the same purpose. The core way in which they function is similar, and yet there is so much diversity in the details, right? So this variation is an evolutionary biologist's joy, right? Because if there's no variation, there is no change that's possible. So you need variation. So variation is what typically an evolutionary biologist would start with, to say, okay, how much variation is there? Why is it there? Is it adaptive? Maybe hopefully one or two times in my talk, is that not everything you see around you is a product of adaptation. And that's very important to remember, because our instinct is to find explanations for things, right? And so anytime we see a peculiar behavior or a particular phenotype, like these eyes, for example, the color of the eye can be a phenotype, we are very tempted to try and say, oh, that color must be adaptive. Uska kuch to function hoga, right? Which is why it has evolved. But that's not always the case. Sometimes, because of chance, because of what we call stochasticity, these kinds of phenotypes can also evolve, or certain behaviors can evolve, not because they're adaptive, but there are many other reasons why they may evolve. One reason may be, for example, that there are other traits which are actually important for reproduction. Okay, and if in our, because of the way genes function and they're organized, it may be that this one gene which changes, for example, eye shape, is actually under selection, under natural selection, it has an important role to play. Let's say if the eye shape is a particular shape, it allows more light to be captured. Okay, so that's clearly beneficial. We know it's beneficial. We can show its benefit. We can measure its benefit. And as eye shape evolves, adapting to the particular environment, maybe the eye color gene is right next to the gene, right, which determines eye shape. Now, I'm of course making a very simplified example here. This is not typically how eye shape or eye color is determined by a single gene. But if these two genes are very close to each other, you can imagine that as this allele or a mutation in the eye shape gene um, is rapidly spreading in the population because it is advantageous, other genes that are associated with that gene can also get dragged along, right? It's something similar to saying, well, in general, let's say there's a family member who becomes very rich, right? And if they're very close to you, you will also get the benefits of that rich family member, right? You will also be able to sort of, maybe buy nicer clothes or whatever you want. And so because one person is successful, you also end up getting the whole family perhaps improves in its ability to do things that they want to do, right? So similarly, genes can drag each other along, right? Genes can also drag other genes down with them, even good ones or beneficial ones. And so there are all these different dynamics that play a role. And so there are many reasons to expect why adaptation may not be the correct explanation for why a particular behavior or a trait has evolved. Okay. All right. So then I want to actually show you an interesting case of adaptation. This is a beautiful illustration by who worked in my lab for a brief time until this year, and he just makes very nice illustrations. I like using his illustrations a lot. So he's made this illustration of different adaptive features of birds, right? And so this is a roseate spoonbill. I don't know if some of you have been to wetland areas. You might have seen some spoonbills that their beak looks like a spoon, and they're really beautiful birds. And so he's illustrating in this diagram here how their bones have air spaces, right? They're not solid bones like our bones are very sort of heavy, but their bones are lighter because they have more airspace. And this is an adaptive trait because it allows the bird to fly more easily with using less energy and so on, right? Similarly, their different parts of their legs are fused, as you see at the bottom of this picture here. So they have fused bones in birds, which are also thought to be adaptive, and so on and so forth, right? So we can have nice and clear examples of where certain kinds of evolved features of organisms are very clearly beneficial and adaptive. There are still more details to be worked out there, but that's generally something we can go with, right? So then coming to what my lab does. So we do many different kinds of projects, and I wanted to give you first an overview of the kinds of things and kinds of questions that we're interested in. How do we approach adaptation and understanding adaptation? So one of the things we're interested in is here what you see on the left, which is molecular evolution. What that means is trying to understand at the molecular level, at the level of molecules, how evolution progresses, right? So it's a little bit of the mechanism aspect of what I've mentioned to you before. So one of the things we are interested in asking is when there are mutations that occur, what is the effect of the mutation, right? Does a single mutation have a large effect on the organism? So, or does it often not? So here's an example of when single mutations have a large effect. You've heard of sickle cell anemia, perhaps, right? And that has a large impact on your red blood cells. So if you happen to have the allele that causes sickle cell anemia and you happen to inherit 2 copies from your parents, then you're going to be in trouble because your blood cells are not going to be the right shape, and they cannot carry oxygen correctly, right? Or as much oxygen as is needed. And so that's an example of where a single mutation can have a really large effect on the organism. But we think that most mutations actually don't have such large effects, right? Problem is very few people have actually measured this because it's hard to measure. You have to first identify that mutation, you know where it is, right? Which is not easy to do. And you have to then measure its effect on the organism, which is also not always easy to do. So this question has been a sort of an open question in evolutionary biology for a long time. And we're now trying to address this question by measuring in bacteria what is the effect on bacterial growth of single mutations. Okay, so I'll come to that briefly later on. Similarly, we want to ask other features of genes or genomes. How big is the genome? What kind of base composition does it have? Are all of these things important in determining adaptive process? Okay, so we ask questions like this at the molecular level, and we typically use bacteria, or we use bioinformatics analyses using sequence genomes that people have already done work with earlier. Then on the right side, you see this gray circle where I'm describing this area called population genetics and ecology. So what are the kinds of questions we can ask there? We typically do this work with a beetle, Tribolium, which lives in flour, that is atta, and it infests everywhere across the world. It's a major pest of wheat flour or other cereal grains. And we use it to ask questions about how much does diversity matter in a population. So if you force populations to adapt to new kinds of atta or new kinds of flours, how do they adapt? How fast does that process take? How long does that process take? Why does it take longer? Why does it take shorter? How can we make it slower? How can we make it faster? What behaviors of the beetles can help in adaptation to new environments? And the point of doing all this is because it hopefully tells us something about how adaptation works in nature in general, right? Not only to learn about that beetle, which is an interesting beetle, but to also learn more general answers to more general questions. And the third area that my lab works in, which is a more recent and new area for the lab, is to talk about coevolution, or how communities are assembled and how they evolve. Okay, so we do this work with typically communities of bacteria, which live inside the guts of different insects. So here we've done work with dragonflies or butterflies, and also the beetle. And asking what kind of process determines who is going to be in this community, okay, inside the insect, and what is their effect on the insect. In return, what does the insect give back to them? Does it give something back to them? And how does all of this influence adaptation of all the partners involved? Okay, so these are the kinds of broad questions that we address. In the next few slides, I will give you few examples of the kinds of things we do and what we found out. But I think there are a few questions, so maybe I'll pause again and take some questions. Okay, let's see, where is the chat?
So we don't have questions yet.
Okay. I thought,
but yeah, we've got one. We've got one question.
Okay.
Vaishnavi, you can unmute yourself.
Sir, how develop gene future in bird?
Oh, sorry, I didn't hear your question. Can you please repeat?
How develop gene future in birds?
मराठीमध्ये विचारायचं असेल तरी विचारू शकते.
मिस जीन फ्यूचर है ना कैसे डेवलप होता है इन बर्ड्स?
इन बर्ड्स, हो, मला बर्ड्स बद्दल ऍक्च्युली पुरेशी माहिती नाहीये, त्यामुळे मला सांगता नाही येणार एक्झॅक्टली. बट लेट मी थिंक. So yeah, so it's not a question that's specific to birds actually, about how gene features develop and how they would evolve. But maybe let me give you an example. So you know how genes, some of the genes in eukaryotes can be interrupted by introns, right? So one very interesting piece of work that some other people, scientists in the world, have done is to ask how many introns and how many exons should there be in a particular gene, right? And does it matter for evolution, for adaptation? And it's interesting because it does. So you know, the site at which splicing occurs, which is where introns are separated out, right? And the exons are then spliced together to make the mRNA, the final mRNA, which actually gets translated in eukaryotes. People have found that people have asked how these introns are born, right? And how these exons are born. How are their boundaries set? And there are very, very interesting evolutionary processes that occur where different organisms who have similar sets of genes because they share a common ancestor, but those genes have now evolved in different directions. And in some cases, some genes in particular organisms, the same set of genes, have evolved so that they have many more introns, or they have introns of different lengths. And that changes the final product that comes out. So that's a very interesting example of how the features of the gene can change sets of organisms in such a way that they allow different features of the organism to then develop according to whatever is the selection pressure that's acting on it. I know that's a very broad answer, but I think that's the best I can do because I don't know enough about bird genes particularly. Are there any other questions?
Ankita is asking, what does coevolution mean?
Oh, sorry, I never explained that. Yeah, so coevolution means that you have two organisms who influence each other's evolution. Okay, so a simple case of this would be, well, why don't we take the case of coronavirus, right? So we expect that if the coronavirus spreads in the population of the world for quite some time, it will sample some new mutations, some of which may be beneficial, okay? And maybe it is beneficial, of course, for the virus. So let's say there's a mutation which allows the virus to infect more people, okay, at the same time or reproduce faster. We also expect that if this virus continues to infect human populations at some low level, perhaps, for a few generations, we expect that humans will then also have evolved the ability to deal with that virus better, right? So this basically this kind of coevolutionary dynamics can shape the long-term evolution of the host and the pathogen, a parasite and a host, or mutualisms, right? So if there's a bacterium, so you know we all have bacteria in our guts, right? And many of these bacteria are beneficial for us. And we help them also by providing them a nice environment in which they get food and they get to reproduce and they get to be transferred, right? And so that kind of mutualism also is a situation where it has co-evolved, right? Where both partners have influenced each other's evolution to some extent. So that is what is co-evolution.
ओके, म्हणजे थोडक्यात म्हणजे दोन ऑर्गॅनिझम्स किंवा दोन प्राणी किंवा प्रजाती इव्हॉल्व्ह होत आहेत आणि त्यांचं जे इव्होल्युशन आहे ते एकामेकांवरती आधारित आहे. बेसिक. ओके, अच्छा.
Okay, shall we continue or are there any more questions?
Aman is asking, can adaptation be considered as variation? So,
can it be considered as variation? They're kind of very different things actually, right? So as I said, adaptation is either a process or an outcome, okay? Or it can be referred to as a trait, for example, which can be adaptive. But variation is a property of a population, let's say, right? So we can say that in the human population, there is variation in skin color, in eye color, and so on, right? That would be variation. But as you say, in the Indian population, maybe there is some adaptation to tolerating high temperature or tolerating or dealing with parasites that we encounter, much more commonly in India than anywhere else in the world, things like that, right? So they're slightly different things entirely. Does that help?
I guess, yeah, well, you can carry on with your talk. We've got more questions coming in, but we can take them maybe a little bit later.
Okay, all right. Okay, so then let me start giving you some examples of a bit more detail of what other kinds of things we do. So here is where we want to ask what are the gene and genomic features that affect evolution, right? And so we've done this work with bacteria. And on the left here, you see a diagram of a ribosome, which is the green part there, which is moving along a messenger RNA, which is an mRNA, which is, and you see the code on the mRNA, which is copied from our DNA, is being read in triplets by the ribosome, right? And the tRNA, which is in maroon, is bringing amino acids, and the correct tRNA is chosen based on the anticodon that matches with the codon on the mRNA, the triplet. And accordingly, amino acids are joined together to make the protein, right? So as we all know, we generally think of proteins as being rather important to produce correctly, right? You don't want too many mistakes in your proteins because then your cells will not work properly, right? So what we wanted to understand is there are so many different processes and details and genes that are involved in making this very basic fundamental aspect of life for it to work properly. Many things are involved, right? And we've been trying to understand, despite this central importance of the machinery that produces proteins in all cells in life, there's a lot of variation in the different components. Okay, so for example, which codons are used to encode specific amino acids, it varies a lot across bacteria, across humans, across mammals, across vertebrates, a lot, right? So some of them might use, might code tryptophan using ACC, and some of them might use, well, tryptophan is a bad idea, but bad example, but let's say glycine, right? Some may encode glycine using AGG, and some may encode glycine using AGC. Ultimately, the amino acid is the same. Then why does this codon use vary so much? Similarly, there is also a lot of variation in the base composition. So is the mRNA largely composed of Gs or Cs, or As or Ts? That varies a lot across life forms. What kinds of tRNAs do you have in a cell? How many of each variety? What kind of sequence modes are used to recognize binding of different proteins? So all of this is important for the function of this machinery, the correct function of this machinery. But each of these components is very variable across life forms. So we've been trying to understand how this variation comes about and what effect does it have on the process of translation, as well as the ultimate evolutionary dynamics of translation machinery as a whole. And so I guess the analogy I'd like to give you here is, imagine you have a very complicated machine, right? Or a toy. Imagine whatever the most complicated toy that you have ever had in your childhood. And now think of all the different parts that could break, right? Or that could change a little bit. So if you change, let's say you have this one machine, one toy where you have to, use that tabi, and then you have to wind it up, and then it walks on itself by itself, right? Now, even though it's a very simple toy, there's actually many different parts involved, right? And so now let's say that or it changes its shape a little bit. So now you have to push extra hard to make it turn. Okay. And the question is, if this is a analogy for the kinds of machinery we have here, many different parts have to function just perfectly. If you change one part, other parts have to compensate. Something else has to change to still have the whole thing function, right? And that is what we're interested in. Given that there's all this variation, every time there are some mutations that change how this precise protein is going to function, something else has to change to accommodate that, right? And so this is the kind of coevolution, in some sense, of different parts, which we're interested in asking, how has it evolved? And so we do that using experiments in the lab, as well as working with bioinformatics analyses. And so what we found out, and this is a hypothesis, we have a broad hypothesis that we've come up with, which we're still testing and we continue to work on for a long time. So we're saying that base composition is something very fundamental, which changes over time, and we've now measured and mapped how it changes over time. How much does it change in different bacteria? But we're saying that, okay, the base composition, for some reason, let's forget about it for now, changes. Once it changes many different things in the cell. Okay, it changes codon use, changes tRNAs, changes mRNA motifs. So we measured the impacts of GC content on all of these things. Okay, and this is what I'm showing in the diagram on the right here. Once these things change, translation rate and accuracy, which is how correct it is, also changes. And as a result, growth changes, right? So ultimately, selection, natural selection, which is not a thing, but it is kind of a process and an outcome both, is not acting on the detail of the tRNA. It is acting ultimately on the survival or the reproduction of the organism, right? And in bacteria, we typically measure it as growth rate. Time, how many more bacteria produced, right? Given the number of bacteria that existed in the past. So if it doesn't affect growth, which is a conglomeration, a combined effect of survival and reproduction, then it doesn't matter. Any changes I make to tRNA that don't change, survival or reproduction of the bacterium, it doesn't matter. It will change randomly, and there's nothing to be understood or nothing to be gained in some sense. Yeah, or there's no direction particularly to evolution. So we think that if we can now understand in a little bit finer detail how these different processes shown in this diagram connect with each other and what are the outcomes, we can better understand how or why these different components of translation change so much or vary so much across different species in bacteria. And I'd like to now give a counter example of this. So everything saying, well, the goal should be to reduce mistakes, right? So because you can't afford to make mistakes, you can't have too many proteins in your cells which are not functioning properly because they have mistakes. But we had an interesting finding where we asked, okay, actually, maybe in some cases, making mistakes is not such a bad idea after all. So we explored this in bacteria, and we find actually that, so this mistakes in protein is loosely called mistranslation, right? So there can be many kinds of mistranslation, but for now, we'll just say that mistranslation is referring to mistakes made during protein synthesis, right? That means there are wrong amino acids, or the protein is the wrong length, and so on, okay? So what we found is, if we have strains of bacteria which have greater mistranslation level, they actually survive better under stresses. And let me draw you a cartoon of this, right? So on the top here, you see a population, which have low level of mistranslation. And the bottom, I'm showing you population, just circles, with high level of mistranslation. What we've done here is we've artificially, increased the rate at which they make mistakes, okay? And now we're going to monitor what happens to these populations with time. And we give them some stress, right? For example, DNA damage. We put them under UV, or we give them antibiotics, which cause damage to the DNA. And what we find is the early survival of these cells without any mutations, before any mutations happen, is different. So populations or cells which have low level of mistranslation survive less, many more cells die. And populations which have high level of mistranslation, many more cells survive, okay? And as a result, because there are many more cells, many more mutations are possible. Some of those mutations are going to be beneficial, okay? Which allow the stress. As a result, in the end, through this gyrase A mutation, in this particular case of this particular study, we find that the population of cells which has a high level of mistranslation is larger at the end of the stress, okay? Which is counterintuitive, right? Earlier we thought, well, mistakes should just be avoided, they're bad. But here it turns out that mistakes are actually good for these cells under these conditions. And so we then ask, well, why are they good? And here in a nutshell, again in a cartoon form, is what we find. We find that normally bacterial cells, when they get stressed, they induce a response called a SOS response, okay? So it's like basically many different functions in the cell change, and the whole focus in the cell becomes, how do we deal with this problem, and how do we get past it, how do we survive, okay? So if I show this SOS activation as this threshold value, this curve, what we find is, we think the wild type cells, which is in the black ball, they are, let's say, some distance away from the threshold, okay, of activation. And the mistranslating cells, which is where we force cells to have a higher level of mistranslation, are a little bit closer to that threshold. So now, when the damage occurs or stress is introduced, the mutant cells, the mistranslating cells, are quickly able to go up, induce the SOS damage, repair it, SOS response, and repair the DNA damage faster. Because of that, before any mutations can occur, these cells are doing better already. They're surviving better. So that is our hypothesis. And just for fun, I'll show you this cartoon, which Pranjal again drew for us. This is sort of, suggesting how this works. So you're seeing these on the top panel in this cartoon, you see these Rishi Munis, right, who are doing, I don't know, they're doing their meditation or whatever. And on the right side, some of you have heard of Durvasa Rishi. This Rishi would always be very angry, right? And that's the two guys on the right. Okay, so they're already very stressed and they're already very angry at everything. Now there's a demon that comes up, okay? And when the demon comes, because Durvasa is already angry, he just throws sharp after sharp, right? And the demon is killed. But the other rishis who are peaceful and calm and just being normal, they die, unfortunately, right? So this is the situation, we think, where being making more mistakes, being more angry can actually be beneficial in certain situations, like when there's a stress or a demon who's going to try and kill you, right? Maybe I'll pause here and ask if there are any questions.
If anyone has any questions, please raise your hands. Mm,
Okay, I guess we can continue then.
yeah.
Maybe this was a bit too much detail, I'm not sure. Um, okay, all right. So I'll go on to another example of the kind of work that we've done. So like I said earlier, we wanted to ask what is the effect of a single mutation, right? And really very few people have measured this. So we to do this, we need to first isolate single mutations, right? And so what we did here is we took agar plates. Maybe some of you who've done some microbiology would know about this. Just simple bacterial agar plates on which we can put bacteria and they will grow. And so that's the cartoon that you see here on the left. So each yellow dot is a single colony of bacteria formed originally by a single cell, which kept multiplying, and formed a colony, right? So we randomly pick one colony every day, and we allow it to transfer, okay, and to grow again. And what this allows is that without much natural selection, because there's no competition for food, nothing, one colony at random is chosen. Okay, so there are mutations which are happening in these population, which they will happen, and they occur randomly. We know this. At some point, if I keep this process for a long time, for many generations, I will find that some of the colonies will have some mutations, right? And because I'm reducing how much selection is acting on it, most mutations, as long as they don't kill the cell, will survive. Now we can take these colonies and we sequence their DNA, okay? And we ask, compared to the original ancestor, do we see any mutations? And because bacterial genomes are relatively small, we can do this without spending too much money on this, okay? And so we sequence the genome and we find, okay, here is one cell here's one colony which had one mutation only. Now we take that colony and we measure how fast does it grow, that is its growth rate in different environments, okay? And the question, we can ask many questions with this kind of setup. One question I want to tell you about is how often do mutations cause trade-offs? Now, what are trade-offs, right? Maybe some of you have heard this, but I'll just explain. Trade-offs means that you can't be good at everything, okay? Now, that is something that most of us know, right? If you want to be good at studying, you will not be good at the latest pop culture reference. You will not necessarily be always good at athletics, right? So now my point is you have limited time or energy, and you have to decide where you're going to allocate it according to your interest or whatever. So that is a trade-off, right? That if I have to really focus on something, spend a lot of time on it, I will not be able to focus on everything that I want to do, right? I will have to give up something. That's fundamentally what is trade-off. So organisms also face this a lot, right? That any organism in the wild, a cheetah, whatever you're thinking of, has limited time, energy, effort, but has to do many things like surviving, reproducing, parental care, maybe moving from place to place. So how do you manage that? And these trade-offs we think are very common in organisms. So how do they arise? And one hypothesis has been that trade-offs arise because a single mutation affects two different functions. Okay, so imagine that in the case of these bacteria, a single mutation could increase the ability to grow very well on glucose, but reduce at the same time the ability to grow on lactose. Okay, the hypothesis has been that such mutations are very common. That very often you'll find mutations that change in opposite directions, two different traits, and that is why we see trade-offs. So we wanted to test this idea. These guys with single mutations, the colonies, we asked how do you grow in different environments, and we asked how many times do we find that mutations actually lead to trade-offs. And surprisingly, we find that it's a very small fraction. And this is called antagonistic pleiotropy. For those of you who are interested, I want to read more. And so this was the first time that we could actually measure how often can single mutations give rise to trade-offs. And the answer is actually not very often. So trade-offs occur, but they occur because of some other reason. Okay, so I should have set up this cartoon earlier. So this is a cartoon we made for a popular article which we wrote about our research. And so this is showing how this one woman who's running on the left, she's a runner, an athlete. she's thinking she's a sprinter, right? So she runs 100 meter sprints, and she's a very good sprinter, but she wishes she could run long marathons, right? Whereas the lady on the right here, she running 10-kilometer marathons, and she says, I can't sprint like her. I can't run at fast speed for a very short time like this other woman can run, right? So that's a trade-off effectively. And we are saying that trade-offs, yes, they do occur, of course, but we think they don't occur because of single mutations. They occur for other reasons. And so now we're doing follow-up work to ask, well, why do these trade-offs occur and how do they come up? So that's another aspect of the work that we're trying to do now. Okay, so then let me go on to a new, another different version of what we do. So we wanted to also ask about trade-offs in other organisms and in natural ecosystems, right? So this is work we did with this flower beetle, which I had mentioned to you earlier. This is a pest species, and so we collected populations of this beetle from different parts of India, which you see here on the map. And we brought them into the lab, we had them lay eggs, and so we built up these large populations. We call them populations in the lab. And now we want to ask, do these guys show trade-offs? How do they adapt? Do trade-offs matter for evolution? And so here's another way in which one can start unraveling adaptation, right? By asking, let's force things to adapt in the laboratory, experimentally, and then go back and say, okay, how did you adapt, right? So we haven't finished with this project yet, but I'll just tell you where we are at right now. So we did this, what's called long-term laboratory evolution experiments to test how does adaptation work. And it's really powerful and it's really fun because it happens in front of your eyes, right? And of course it takes years sometimes, but anyway. So what we did is we took these 10 founding populations, which were obtained from wheat. They were typically found in wheat flour, and they're really good at using wheat. They reproduce really well in wheat. But if you now put them in two different flours, corn and finger millet is ragi, which some of you may know about. So they don't do very well in these flours. They have a hard time surviving and reproducing. So we force them to adapt to these. And so how do we do that? So we collect all these beetles and pupae and larvae, which are the different life stages that you see on the right here. And we have them go through these cycles of reproduction, right? So we let them reproduce for 3-4 weeks. Then we throw out the dead individuals, and whoever is alive, all the fresh individuals, we put them in fresh flour and we do another cycle, right? And so we did many such cycles for the last 5 or 6 years. My students have been doing these cycles. And here is the interesting stuff that we see, right? So what I'm showing you here, there's a lot on these plots, but I'll explain. So on the left, starting from the left, what you see on the y-axis in these plots is number of adults, right? So one simple measure of saying, are you adapting, is to ask how many individuals are there in your population. If that number is large, you're probably adapting well because your population is growing. It's a positive growth rate. And if you're declining, you're not able to adapt, right? So each line here is a single replicate population, because we have 30 such populations. And we're showing you what happens to the number of adults over time. And time is on the x-axis, right? So generation. And what you see is very interesting dynamics, right? So let's first focus on the wheat. And also the two rows are showing you adult size and larvae size, okay? So we'll focus mostly on the adults today, but the larvae are also interesting, as you can see. So what you see with the wheat is initially there's a big boom, okay? Population size increases, there are almost 1,000 individuals, adults, and then they crash, okay? Probably because of what's called density dependence, where they can't survive. You know, there's too much competition. Not everybody lives. Most of them die, okay? And then they reach this sort of more or less stable state, right? Where there's some fluctuation, but surprisingly and interestingly, very parallel dynamics, right? So every population is independently evolving. Some mutations will occur, some live, all of that, and yet the outcome overall is quite similar, okay? Now, this is not particularly surprising because wheat is a good flour for them. They're already adapted. Finger millet is interesting. You see a overall gradual increase, right? So the adult population size increases initially, crashes, and then slowly with some, up and down, it is increasing with time. Corn is also very, very fascinating, and this is what's expected. So in corn, if you notice, initially, there's a large crash, right? There's no boom. And I told you that corn is bad for them. They don't survive well. They can't reproduce well. There's a big crash. and then barely they escape extinction, right? Because look at that number, it's almost zero, not quite zero, but almost zero. And then they start adapting, right? So that's really, really interesting. So that is what is adaptation. That U shape that you see there is what's called evolutionary rescue, where this population has been rescued from extinction. Okay. And now we can ask, well, what are the genes that were responsible for this? What behaviors were responsible for this? These are all the questions that we're asking now, okay, in the laboratory. And in the larvae, you see much more stable dynamics, but I think I won't spend time on the larvae right now. Okay, finally, I think there's one last bit of stuff that we do. We are interested in host and bacterial interactions. So here's just a cartoon showing you how we think many insects will acquire bacteria from the environment, right? And this is not just insects, this is all organisms, right? So for example, every time you eat something, you're not only eating the food, you're also acquiring microbes. And then many of those microbes, depending on your diet, will either establish in your gut or will be thrown out, will be outcompeted by other bacteria which normally live in your gut, or they may colonize your gut, and that is sometimes what is leading to problems of the bowel or bowel diseases, right? Where pathogens or like Clostridium can infect your gut lining and then cause problems, right? And they're not part of your normal flora. So we want to understand this process of how these kinds of interactions between host and bacteria evolve. How do they change? What is the ecology of these interactions? And we do this work again in different ecosystems. We're doing one project with rice varieties in the Northeast. So these traditional rice varieties that are grown in so many parts of India, which are now dying out, unfortunately, we found that they host different kinds of bacteria on their leaves. And so now we're trying to ask whether there's any benefit that they give to the host, and how is their evolution shaped. And we've also worked on different kinds of gut bacteria that live inside different insects like dragonflies and butterflies and so on, right? So that's another kind of work that we do. And I think in the end, just finally some interesting, well, not data, but interesting work that we did. This is a PhD student. She worked on butterfly microbes, right? And so there are two different butterflies you see on the top panel here, the left and the right. And these are very common butterflies. You may have seen them outside even. So the left is called a plain tiger butterfly, and the right you see castor butterfly. So we caught wild females, we had them lay eggs, and we eliminated the microbes because we wanted to ask what happens if you get rid of the microbes in these larvae, caterpillars. So we sprayed either antibiotics on the leaves or fed the larvae antibiotic treated leaves, or we chemically sterilized the leaf surfaces, which is what you see in these cartoons here. And then we asked what happens to the survival and the growth of these larvae. And the prediction was that if we remove the microbes and if they are important, we should see that the larvae maybe don't grow as well, or they die faster or something like that. And so yeah, and here's a agar plates that actually show that our sterilization treatment was working, right? So on the left side is a plate, which is just a normal plate where you see lots of bacterial colonies are growing. On the right side is the plate which is coming from leaves, which we had sterilized or treated with antibiotics. And so in a nutshell, what we find is removing the microbiome does not change butterfly growth or survival. And therefore, it seems like even though the butterflies have adapted to different diets, very different sets of plant diets, they do not involve bacterial partners in this adaptation. And that is surprising because there's a lot of literature, a lot of work that many people have done in the last 10, 15 years, which clearly shows that many cases, most insects, most animals that have evolved to use specific diets or different diets, they all rely on some microbial partner or the other. Okay. And so here we're finding that antibiotics, the butterflies are fine, right? And so that suggests that whatever adaptation butterflies have had to do, the evolutionary adaptation that has occurred as butterflies have evolved to eat different plants, right? They don't use microbes at all. And so we're trying to understand why. And this is a bigger question in general, right? So I'll stop there. This is my lab and the sources of funding that have supported a lot of the work that we do. If you'd like to know more, please do look up the website of our lab and read our papers. And we also publish very often popular research articles or summaries of our research to make it easier to understand without the technical terms. So please read those as well. Many of those are on the NCBS website. So I think, yeah, I'll stop there and I'll take any other questions that people may have.
Thank you so much, Dr. Deepa. If anybody has questions, please raise their hand or put it in the chat box or the Q&A box. Um, I'd just like, I have a couple of questions which I'd like to ask on behalf of the students. आणि
Sure.
पहिला एक प्रश्न आहे की एक असं सिम्पल एव्होल्युशनचे कुठले एक्सपेरिमेंट्स कुठला स्टुडंट कॉलेज लेवलमध्ये किंवा त्यांच्या घरामध्ये कुठले करू शकतात का? कारण की तुम्ही खूप फुलपाखरांवरती, बीटल्सवरती काम करता. तर तसं काय मुलांना त्यांच्या लॅबमध्ये किंवा घरी करायला जमेल का? तुम्ही थोडे एक्झाम्पल दिले तर छान होईल.
एव्होल्युशन एक्सपेरिमेंट्स एका प्रकारचे करता येतील. म्हणजे फॉर एक्झाम्पल तुम्हाला सो एव्होल्युशनरी चेंज कॅन हॅपन either very quickly, okay, within one generation or across many generations, right? आणि हे जे खूप जनरेशन्स मध्ये होणारे चेंजेस आहेत ते ट्रॅक करायला थोडं तुम्हाला इक्विपमेंट लागेल, थोडी काहीतरी लॅबोरेटरी किंवा काहीतरी कंडिशन्स लागतील आणि मी तुम्हाला सांगते काय लागतील ते. पण जी पहिली गोष्ट आहे की जो एका जनरेशन मध्ये मॉर्टॅलिटी जर का व्हेरिएबल असेल तर यू कॅन ऍक्ट, दॅट इज ऑल्सो एव्होल्युशन, राईट? So that you can examine yourself. So for example, you can take different, तुम्हाला वेगवेगळे किडे आणता येतील आणि त्यांना वेगवेगळ्या स्ट्रेस कंडिशन्स मध्ये टाकता येईल, राईट? किंवा तुम्हाला बिया वेगवेगळ्या झाडांच्या वापरता येतील आणि एका पर्टिक्युलर पीएच मध्ये किंवा एका पर्टिक्युलर टेम्परेचर मध्ये किंवा पाणी कमी घालायचं, पाणी जास्त घालायचं, ह्या सगळ्या कंडिशन्स देऊन कुठल्या प्रकारच्या बिया रुजतात, कुठल्या प्रकारच्या बियांमधनं झाडं येतात हे बघणं सहज शक्य आहे, राईट? तुम्ही जर का शेतातनं तुम्हाला काय ते कवच न काढलेले तांदूळ जर का मिळाले तर तुम्हाला त्यांची मे बी सॉर्ट करता येतील की काही तांदूळाचे दाणे मोठे असतील, काही छोटे असतील, काही थोडे वेगळे असतील. काही फरक आहे का त्यांच्यात की कुठले जास्त नाही? तर ही खूप साधे एक्सपेरिमेंट्स आहेत ते तुम्हाला करणं घरी शक्य आहे टू अंडरस्टँड हाऊ नॅचरल सिलेक्शन वर्क्स, राईट? इन टर्म्स ऑफ अडॅप्टिव्ह प्रोसेसेस विच टेक अ लिटल बिट लॉंगर टाइम, तुम्हाला जर का साधी मायक्रोबायोलॉजी किंवा अशी लॅब उपलब्ध असेल तुमच्या कॉलेजमध्ये, फॅन्सी काही लागणार नाही तुम्हाला, खूप साध्या साध्या गोष्टी, साधा मायक्रोस्कोप असेल, साधे तुम्हाला आगार प्लेट्स असतील जर का करता येणार शक्य असेल, त्याच्यावर तुम्हाला एक्सपेरिमेंट्स करता येतील. तर आम्ही जसे केले तसे. फॉर एक्झाम्पल सिम्पली तुम्ही इकोलाय बॅक्टेरिया किंवा कुठल्याही बॅक्टेरिया तुमच्या तुम्ही हाताचा प्रिंट जरी घेतलात तरी भरपूर आपल्या बॅक्टेरिया असतात हातावर, त्यांच्यातला कुठलाही एक बॅक्टेरियम घ्यायचा आणि तो काही वेगवेगळ्या स्ट्रेसफुल कंडिशन्स मध्ये काही दिवस ट्रान्सफर करायचा आणि या एक्सपेरिमेंट्स केल्या तुम्हाला, फॉर एक्झाम्पल एखादा अँटीबायोटिक तुम्ही घेतलं आणि त्या अँटीबायोटिकचा डोस तुम्ही फिक्स केलात आणि दररोज थोडं 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 कल्चर ट्रान्सफर करायचं stress, at some point later you're going to have bacteria evolve which are antibiotic resistant, right? And so that is adaptation, of the bacteria to the antibiotic, and that is exactly the process that happens in our bodies, right, when we take antibiotics. And maybe तुम्ही ऐकलं असेल अँटीबायोटिक रेझिस्टन्स हा मोठा प्रॉब्लेम आहे भारतात आणि सगळीचकडे जगभर, कारण अँटीबायोटिक लोक खूप जास्त वापर करतात आणि बरोबर प्रकारे वापर होत नाही त्याच्यामुळे हे प्रॉब्लेम येऊ शकतात. तर ही प्रोसेस आपण साध्या स्वरूपात अगदी सहज शक्य आहे लॅबमध्ये करणं. सो तुम्हाला अजून स्पेसिफिक डिटेल्स पाहिजे असतील, आयडियाज हव्या असतील तर मी नंतर तुमच्याशी बोलू शकेन किंवा मला ईमेल करा, मी तुम्हाला अजून सुचवू शकेन काही गोष्टी. चालेल?
Thanks so much, Deepa. We're also running out of time right now कारण की 1 वाजलेत आणि मुलांचे वगैरे पुढे लेक्चर पण असतील. तर मी लॅबची लिंक चॅट विंडोमध्ये टाकली आहे. तर any of the students, you can copy that link and you will find all the information of Dr. Deepa Agashe and her work there. And if you're interested, you may be able to contact her or email her, and maybe you can do some fun experiments as well. She is always there to talk to you. Another thing is, I'd just like to thank you, Deepa, for doing this talk. It's a big experiment right now. What we're doing is, going out and having these conversations, which have not happened before. And I'd like to thank Dr. Rashmi More also for being on board on this idea so that we can do these things. So Dr. Rashmi, if you want to say something now.
Yeah, sure. Okay. On behalf of Pune District Education Association's Professor Ramkrishna More College, I would like to thank Dr. Deepa Agashe for sparing her valuable time and sharing her knowledge and her work in the area of adaptation, and for covering all the aspects of it, like ecological and genetic base of adaptations, reasons for mutation, by providing examples from diverse animal groups and the work that is going on in her lab. Thank you once again, ma'am, for enlightening my students, and we look forward to see you again for such more lectures. Thank you.
Sure. Yeah.
Thank you, Mahin Khan, for joining us, and very special thanks to Chandrakant for taking initiative in organizing this talk. Thank you to my students from Department of Botany and Zoology and staff as well for your active participation. Thank you.
Yes, thank you so much.